hungry. But it's very important that you know about the different steps and aspects of a scientific investigation. You will surely get questions on scientific investigation, hypothesis, etc. So while I've said the word hypothesis, let's get started. What is an hypothesis, Colin?
Well, first of all, if you formulate a hypothesis. So it's an attempt that you explain an event or an observation. First of all, it's a problem that you see somewhere and you ask yourself this question. And then you try to formulate this hypothesis according to that problem to see a possible outcome of what you can expect.
Yes. In other words, hypothesis is, I almost want to say, an informed answer or guess on a scientific question. A question is asked, and then we can say if we ask a question such as, in which type of water will the tea steep, or will the tea bags dissolve more easily? Then I can say, okay, I want to do an investigation to determine that. And then I can say, my hypothesis is that the hot water will make the tea steep faster. In other words, the tea will become tea more quicker in hot water. So you get the idea. And then it is so important that when you write an hypothesis, it can be tested.
Yes, and exactly what you just said. You took a tea bag, you decided, I'm going to take cold water, I'm going to take hot water, and you are going to observe the outcome.
Yes.
That means that either at the end your hypothesis is going to be supported, or it's not going to be supported.
Yeah, we will find in textbook that it says, as on the slide, that it will be accepted or rejected. The trend now is, and make a note of that, to rather say that your hypothesis will be supported by the evidence, or it will not be supported by the evidence. Right, so let's continue. What other information do we have here? So if we look at the hypothesis that you will get marks for, this is what it must have.
You must have your two variables, a dependent and an independent variable. So today we're going to give you also that opportunity to distinguish between the two and know exactly how to identify the dependent and the independent variable. So please make notes as we go along.
Yes, we've only got about 10 minutes to talk about this. So unfortunately, we might have to rush a little, but please make your notes and you've got your hand out. So you've said, Colin, that the hypothesis must have two variables, dependent and independent. And you must state the relationship between the two. In other words, it's like cause and effect, right? Your independent variable will result in then your dependent variable.
And when you say there's a relationship between your two variables, that is either there will be let's make a simple example and say there will be if the one, that's now the cause. Say, for instance, we take water, the amount of water. If you say more water will allow seeds to grow faster or higher, because how do you, remember it must be testable. So your result that you want to look at should be that we can measure it at least, or observe and measure, for instance. So that is what you must always look at at what and how to state that your two variables.
Now, before we get to those variables, it is very important that we have controlled or fixed variables. These are the things that we keep the same throughout our investigation, so that we can say the results of our experimentation, our investigation, are valid. In other words, it is believable. There was no influence from the side that can do something. In other words, if we say, what is the factor that will cause seedlings to grow faster? do we give a lot of water or do we give little water? Those seedlings must all grow in the same type of soil, because some soils may be more conducive to growth of seedling. It might contain more nutrients. They will all have to grow in the same type of soil, in the same sized container, with the same sunlight, with the same temperature.
But also important, Lorraine, the same amount of seeds.
The same number of seeds. You can't say, okay, these grew faster, look at the lot. And then you planted 50 seeds, but only 5 seeds here. Oh, no, only 5. So I think you get the picture. It is so important that these fixed variables will ensure the validity of the investigation.
Yeah, and we'll get to that word again, that term.
Yeah.
Let's go back to our independent variable. Which one is then the independent variable? Now, that is the factor that we want to investigate. Now, like any other investigation that we have, there's a normal procedure that we follow. And that is why we say we're busy now with a scientific investigation. So we're taking a procedure that is a recipe that we work according to, and we change it into something that we can investigate and observe changes.
So the independent variable is the one that you decide to manipulate to change. In other words, you decide, am I going to give the seedlings 10 milliliter of water, or 5 milliliters, or 2 milliliters? And the dependent variable then, Colin, is?
That is normally,
yeah.
It shows us, it refers to the effect that 10 milliliter or 5 milliliter or 2 milliliters water had on the seedlings, the growth of the seedlings. So there we have, this the effect of your independent, the one that we can measure at the end of the day. If we want to see how many seeds eventually have germinated, then we can say we started off planting 20 seeds, and with 2 milliliters of water, only 2 seeds germinated. And then we can say with the 10 milliliters of water, we saw 12 seeds germinated. So it's something that is measurable. You can count, you can either, or when it starts growing, which one will grow faster. You can measure the height of the stems as they come and grow above the soil. So that is your dependent, the one that you observe, the one that you want to measure, that is your dependent variable.
Right. And at this stage, we would ask you to please ask your questions if it refers to the work that we're busy with. I can see there's somebody either from George or called George logged in. I can see Matthew Guniwe School, welcome everybody, and then 2 others without names. But please tell us and your questions, ask your questions. If it is relevant to what we're doing, we will happily answer your questions for you. There's a very important note that you made there.
And Lorraine, I think you should emphasize that, please, because year after year, when we mark, we see that children don't follow this one.
Somewhere in the question that you will get at the end of the year, and also in June, the June exam, and also in the September exams, you will get a question on a scientific investigation. And they might tell you something like a group of scientists decided to investigate, or a learner wanted to find out, That is the aim of your investigation. In other words, what are you going to do? What are they going to investigate? An aim starts with to determine, to find out, etc. And from that aim, you can formulate your hypothesis. From that aim, you can identify your independent variable as well as your dependent variable. So go and look for the aim of the investigation. And I promise you that because both Colin and I mark matric papers at the end of the year, the memorandum will always take the hypothesis and those 2 variables from the aim of the investigation. And then something else, and Colin, we will get to that later too when we do the questions, is that I cannot there was one year a question about which type of chicken grows faster. The one that was grown to give more meat, or the one that was bred to give more eggs per day. And now I can't look at them and say, this one is growing faster, that one is growing faster. So I can only put them on scales. So the mass is an indication of the growth. It is how we measure the dependent variable. But I think we'll get back to that again. Right, you said we were going to talk about reliability again.
Yes, you touched on the validity part quite well, but I think we'll do it again.
Yes, we must.
So, reliability, and I emphasize the R of the reliability.
Roll the R for reliability.
Yes, I have to do. So, what makes it more reliable? Any investigation, or you can also say that what is it that you haven't done that maybe is not going to make your investigation as reliable? And reliability means that the results that you get, if you do it more than once, that means if you repeat, and that is why I emphasize the. So when they ask you, how can you increase your reliability? Remember, you can repeat the investigation. But also depending on, we spoke about something earlier, but maybe when we get to that question, we can refer to that. Remember, there are certain things that you can repeat, but there are certain circumstances that you can't repeat.
Just to explain to them, I'm just over 1.5 meters tall. I'm not going to give my age, but if you have to give an investigation about women, how tall women of my age are, and you just look at me, you will say, okay, 154 centimeters, and that's it. But if you look at, okay, Colin is a bit younger than I am, but if you look at other women of my age, you will find that there might be women of 2 meters and there might be women even shorter. And the more women of my age you look at, you will find that there is a huge variety, so that you can get an average, which will be much more reliable than just looking at me. It might be a stupid little example, but I think you get the idea.
But in your little handout there, you can see there's an answer, possible answers that you can give when they ask you, how can we, for instance, improve the reliability of an investigation? First of all, repeat. And then, of course, whatever the amount you're taking to test, increase that sample size.
Good.
Sometimes they don't understand the word sample size. It is how many am I using? Like I said, instead of using 20 beans in our investigation, now we can increase it to 40 beans for each. So it is repeat, but also increase your sample size. And here you come to your validity again, Lorraine.
Yeah. In other words, people want to know, but did you do this in a way that these results can be trusted? It's not just doing it the correct way and taking a big group where we improve our reliability of our results. It's also, this is about how we did the investigation. Was the amount of water added to the seeds the only factor that caused the seeds to germinate? Remember, I said in the beginning it must be in the same type of soil, in the same amount of soil, the same number of seeds in each of the containers with the different volumes of water. In other words, whoever reads my report, about my investigation, wants to know that nothing else influenced this investigation. In other words, that when I said it was the amount of water that caused different numbers of seeds to germinate, whoever reads my report can believe, yes, it actually was the amount of water and nothing else. And that is important for a scientific investigation, because now people can believe and they can go further and they can test more aspects because they already know what the influence of water is.
And as you see there, all the factors that you must control or keep the same, except the variable, and you've listened to Lorraine what she said, the different amounts of water that was not kept the same. So that is the factor that we were actually investigating.
In other words, if we want to know whether the soil types have an influence, then we give equal amounts of water, but different soil types. We only test one factor at a time, only one independent variable at a time.
And there's a possible answers on your notes there. You can see which factors might have decreased the validity of an investigation, and that is some factors. And look at the question and name those things in the question that you can see that should have been kept or be controlled, that was maybe not controlled. So that could have decreased the validity if those things were not kept fixed
And
or
you
controlled.
know, Colin, it is so important when they get to questions. They will tell them that the scientist went out to count the birds at 7 o'clock in the morning. And they counted the birds by standing on the hilltop and seeing how many birds flew past. I'm just grabbing here random examples. Now they ask you, which factors are your constant factors? Then you must say, they went to measure. You can't say the time. What about the time? They counted the birds at the same time every day. The method. Now, what about the method? They counted the birds in the same way, with the same method. They did it at the same time and the same place every day. So you need to tell us that the amount of soil was the same. The number of seeds was the same. Do you understand? You can't just say seeds, soil. You have to give an explanation. Sometimes you have to mention in a sentence. If they say mention, it doesn't only mean a word always. Good.
We're just going to skip the question on investigations because we're running out of time a bit. Let's get to protein synthesis, and when we have enough time left, we can always come back because it is all the answers. The memos to those questions are also in your workbook. So let's look. Proteins, where did we hear about proteins and where did it start in your curriculum?
They must switch quickly now.
Yeah. So there you see, it started in grade 10. In grade 10, when you did the cell structure, we started looking at the nucleus. What was found in the nucleus? Where was RNA found? What is the function of the RNA? And what were the functions of the ribosomes? And those were all the stuff. And then additional to that, you also started when you did organic substances, because there you learn that the monomers of proteins are amino acids. So all the little things that we need to understand protein synthesis started in grade 10.
And they even did it in primary school. And I've just finished talking about the cell with my grade 9s. So yes, they should know all these basic things by now. And they should know the structure of a cell, where photosynthesis occurs, and how it occurs is what we are quickly going to talk about now.
Right, so proteins are then produced. Synthesis means to produce or to make. And the process there, it's made, and all enzymes are proteins. Most hormones, not all of them, are also proteins. But these, the building blocks, the smallest units that a protein consists of, are our amino acids. And here we must also know that DNA and RNA are involved, and there are 2 stages that we are going to look at.
Yeah, and my learners always tell me they can never remember which phase or which stage first. They must actually go and learn. Now, the 2 stages are transcription and translation. And if you think transcription means to write over, and translation, if you look at the literally meaning, that is to take from one form to the other. So you first write, and then you translate to somebody else what you wrote. So think of that: writing, transcription, and then telling, translation. So maybe that helps, a little bit of an association, so that you can remember the 2 processes in order. Colin, where are we in the cell?
Right, now that we are busy with the protein synthesis, where in the cell are proteins made? At the ribosomes. Where do we find the ribosomes? Outside the nucleus of the cell, in the cytoplasm. Now, those proteins are made, and there is a certain instruction for which protein, just ignore the second N there, where in the cell is the code, that instruction for a specific protein to be made. That is in the nucleus. So we have a problem.
They learned everything is controlled by the nucleus. Or how can the nucleus do it? Because it's got the DNA inside. So for all proteins, the code of which protein is formed and what it looks like is in the DNA. And did you hear Colin said we've got a problem? It's in the nucleus.
Yes.
And where does protein synthesis take part? Outside the nucleus at the ribosomes. So your solution?
The solution is, first of all, before we get to solution, the problem is the code is on the DNA. But the DNA can't leave the nucleus.
Exactly.
Because it's too big. It's a huge molecule. So how does that instruction get to the ribosome? And this is where we get in the help of mRNA and tRNA. So there's our link. mRNA is the link between the nucleus and the ribosome.
And what is mRNA?
It stands for messenger RNA.
Yeah, there you've got it. So, in other words, mRNA, the messenger RNA, takes the message from the nucleus outside of the nucleus.
To the ribosome.
Right.
There we go. There I have a little diagram for you, but I'm going to cut it up quickly for you.
That's a cell, yes.
Yes. Because this is the full cell. There is the nucleus, and that's the rest of the cell. But let me explain the process, because you need to know the process. And the first one, like Lorraine says, is that transcription must take place in the nucleus. Transcribe, to rewrite. What must be rewritten? Now you must understand the whole DNA is a big, big molecule. And the DNA is in the nucleus. But the whole DNA strand, that double helix, can't unwind the whole thing. Otherwise, another type of process will take place.
You see, what happens here is that the codes, or a code for a protein, is found on a section of DNA, a gene. A gene will code for a specific protein. It's only a section of DNA. The rest of the DNA is not involved. And that is when you look at the second picture again, like Colin said. It's only part of the DNA molecule, the one where the need for a specific protein is. Now, we're talking about a protein, but we haven't even got a messenger RNA yet. So when this DNA unwinds, what is the following step?
Okay. But for those learners who are really struggling, we're going to make it as easy as we can for you. First of all, you will say that the double-stranded helix unwind. But now this is a little bit confusing because it's double-stranded, but all of a sudden we see 3 strands here. Don't look at that. There's the outside line of the one strand, and there's the other strand of DNA. That is the mRNA that must write the code. But now look what they say. It unzips. Can you see it opens up? And the 2 strands, they separate. But you can see there's only one strand of M or messenger RNA because only one of the 2 strands, that strand is not being used. Only the bottom one is used as a template. And the template is the code. And mRNA, can you see, has now written the code from the DNA. That is the mRNA, and here we have enlarged it.
In other words, what you're saying here is that the one strand of DNA acts as a template for the formation of a section of mRNA. Good.
So now you must be able to describe how is mRNA formed. Now, in your nucleoplasm, there are free RNA nucleotides. So these free RNA nucleotides come, and according to the DNA, now if there's our RNA, you can see every 3 bases form a codon. That codon was written from the base triplet of DNA. So DNA has the code. And the code, it's called the 3 nucleotides here, it's the base triplet. And mRNA will rewrite that code in RNA nucleotides that is found in the nucleoplasm to form a codon. Now, I wonder, are our learners, are they connected? Do we see any messages from them?
I've got a few connections,
Okay.
but not messages
Not
yet.
messages, yes.
Yes.
So if the messages, I wonder if they can give us, now you have to work back. And we're going to tell you how important that is. There we see the codon on mRNA, that first codon, the code is UCA. So what was the original code on the DNA? So we know that DNA does not contain any uracil, but instead it has thymine. So uracil will then pair up with adenine. We just go with the letters now because we know C stands for cytosine, with G, guanine, and adenine with thymine. Don't get confused. All you need to write down at the beginning is DNA does not contain any uracil and RNA does not have any thymine.
That's one of the differences between
Yes.
the two that they must know. I want to draw something. Shall I do that?
Yes, I'm going to quickly go and complete this.
Yes.
And you have in your guideline document an exact description of transcription in your guideline document. Please follow that. But can you see all those words that I've highlighted? Those are the words that the examiners are looking for and the sequence of events as it take place during transcription.
Colin, you will remember last week on Thursday, we had a session where we discussed the outcomes and all the problems and all the good things about the National Senior Certificate exam of 2018. And one of the things that was said was that our learners must use the scientific terminology. Words such as transcription, unwinds, template, free RNA nucleotides, complementary. But can I just quickly, because
Sure.
it's time, okay, what we have is then a code on the DNA. And that code could be ATC, 3 bases, nitrogen bases. And it is transcribed into a codon. And this is why I'm writing, so don't get confused. Code on the DNA, codon on the mRNA. Now the matching codon for this code, A will bind with T, I hear you say. But because this is mRNA, A will bind during the formation of mRNA with the uracil. T will bind with A, yes, and mRNA does contain adenine. And C, you know, with G. So there's my codon. My codon will be, and Colin will explain that now, translated into an anticodon, which is the tRNA. And the anticodon will match the mRNA. T will match U in bringing an A. What will match with A? Not a T. Because we're busy with RNA, it will be a U. And with G, a C. Can you see the similarities, although they're not the same? And so we've done this part, and now we're going over to translation. We've written the codons, we've transcribed the code into a codon, and now the codon is going to be translated by the anticodon.
Before we move away to that, maybe we can have the whiteboard again, just so that our learners know. The code is on the DNA, the instruction. mRNA rewrites that message into RNA, take it to the ribosome, so that the anticodon, which is on tRNA, must now translate that message into what? Into a protein. So the tRNA, and now we can go to the slide, each tRNA looks completely different. Look at the mRNA, it's a short strand. The tRNA only consists of 3 bases here, 3 nitrogenous bases, and it forms the anticodon. And your anticodon must match the bases on the codon. So, here you see there is a tRNA bent backwards, and at the end it carries an amino acid. And that is what we said, that each tRNA carries now a specific amino acid. Why do we say specific? If you look at all the codes on the DNA, that means it's actually the DNA that determines that amino acid at the end of the day. And that is
via
why
the mRNA, via the tRNA. And can I just interrupt, Artie? I hope we've answered your question now. And thank you, because she asked to explain how the tRNA is formed. And it is in the cytoplasm, and it's part of the whole process. And it is there, folded back upon itself, with the 3 free bases.
Yes. And as I said, that anticodon must match the codon on the mRNA. And this is exactly how if you have to describe the process, this is exactly in the sequence of events that take place. And then all the amino acids, as the tRNA come and they lock on with the anticodons to the codon of the mRNA, they drop the amino acid that they code for, because each one of them has a specific amino acid. So the sequence of the amino acid, let me explain to you. We have 10 digits from 0 to 9. And with that combination of those numbers, you can call anybody's number you can dial.
In the world.
In the world. It all depends on the sequencing of those 10 digits that you get out to a specific person. So we have 20 amino acids, known ones that we work with here, and it's the sequencing that will make different proteins all the time, just like your number and your mother's cell phone number and your friend's cell phone number. So each one has its unique number according to the sequencing.
And the same combination will always give you the same person on the other side. Well, unless somebody's stolen your phone, it will always give you the same person on the other side of the phone. The same codes, codons, anticodons will code for the same amino acid. And the same sequence of these base triplets, the codons and the anticodons bringing the amino acids, will each and every time give you the same specific protein. And that is so important, because proteins, if we really have to choose, Colin, we will have to say that proteins are probably the most important type of molecule. And then we can't ignore the others. But let's rather say that the number of functions of organic molecules, nothing comes close to proteins. So that is why it is such a carefully coded process.
If you think about it, over 99% of our DNA codes for proteins.
Exactly.
That is how important it is. The way we sit here, if we just look at one another, all the structures that consist was made up of proteins. So yes, protein synthesis is quite a big thing, and you need to know it. Remember, it's one, as we call it, a big idea. You won't find one question paper 2 without protein synthesis. Remember, DNA, RNA, there are many other things that we could have talked about and spoken about now. But let's just take the protein synthesis, because up to 20 marks can be on protein synthesis.
And last year, the long question, the essay question was on protein synthesis and the types of RNA involved. Now, you will get one. It doesn't mean that it will be the same type of question. Now, might be a section B question or A or both. Can I just go back, and while we have 2 seconds before we start with the next one, Tabile asks, how are the 2 strands of DNA held? And I assume, Tabile, you want to know how they are held together. And remember, we said they are held together, the 2 strands of DNA, those nitrogen bases, by weak hydrogen bonds, which is really rather a force of attraction. That is why it's so easily broken during DNA replication or during transcription. Colin, can I just...
Yes, while you look at all the questions coming through, let me just look at the slide again to end of translation. And as the amino acids, as they attach to one another, they are attached to one another. And please, you have just spoken about a bond, the hydrogen bond that keeps the two nitrogenous bases, for instance, together. But this is also an important bond that appears in most question papers, and that is those amino acids that they are, maybe they stick together, but they bond by means of a peptide bond. Know that bond as well.
Don't get confused.
So the sequence is determined. First, the code was on the DNA. The mRNA rewrote that code, brought the message to the ribosome, and that sequence of the amino acids is then determined by the codons on our mRNA. And as the tRNA brings them to the mRNA, they attach and form this polypeptide until the protein is completed. Once again, that second stage that is in the cytoplasm outside of the nucleus, it's also in your guideline document. Go through it. Look in the mind the gap. There is a beautiful diagram. Those that I've cut up into smaller pieces for you, there's a full screen for you and on how it looks. Go through the mind the gap. Look on the WCED's webpage for matric help and assistance, and you will find many resources that you can make use of.
Right, the questions that we have on the section that we just did. Sia Bolele from Noorderpool asked, why is it that the DNA has the same sequence, but at the end, the amino acids, they are not the same? Remember, you've got billions, literally, billions of nucleotides on one DNA. This section codes for that sequence of amino acids. This section for that type of protein with its amino acids. So that is not a problem.
To give them an indication of how big the gene, how big the gene is on the DNA, your smallest protein consists of at least 50 amino acids. Now, if on the DNA, the base triplet consists of 3 nucleotides for one amino acid that codes for one amino acid. That means if the protein consists of 50 amino acids, that means there must be 3 times 50 nucleotides that eventually, that is how long that strand is. So 150 nucleotides, that will be the shortest strand of the gene that will code for the smallest protein.
Okay, let my friend just hang on for a moment. There's a question from Crestway about could we explain translation again. Unfortunately, we don't have time, but make sure that you have the handout from the department for the telematics. And if your teacher does not have it yet, he or she should contact the subject advisor, and it will be provided. What's the difference between peptide and hydrogen bonds? Peptide bonds is a chemical bond that holds the amino acids together, where they bind by means of peptide bonds to form the protein. A hydrogen bond is the force of attraction between the nitrogen bases of the 2 strands of a DNA. And there was one last one that I wanted...
While you look, let me just explain to you, these bonds don't just break or attach. They are all controlled by enzymes. And the names of the enzymes you don't have to know. Just that you know that it is controlled by enzymes, please.
Yeah, and then, as I've said, we've had Percy Mandala, Nompumelelo, and there was a question for the accounting gentleman, so unfortunately we can't pay attention to that. But yes, how are anticodons formed? They are formed by chemical processes in the cytoplasm, and the process by which mRNA is formed is transcription. Tabile, you go and have a look. Transcription and translation. You need to know those names. You need to know the processes. And Colin, I'm going to shush now so that you can start with our last part of the presentation, mutations.
And if we have a little bit of time, we can still go back, but
Yeah,
we're
but
not
let's
going to
just do
promise that
mutations.
because you have your answers in your little workbooks. Let's just understand mutation. What is a mutation? You must be able to give a definition or define what a mutation is. Now, a mutation is very simply said, it's a mistake. So it is a change, and I'm only going to do with you, and we're only going to do with you, a gene mutation. Because when we are going to look at meiosis, we're going to look at a chromosome mutation. So,
yeah, it's the only two that you have to know.
Yes.
A gene mutation and a chromosome mutation. So today it is the gene mutation, a change in the gene. And then when we talk about meiosis, it will be a change with the whole chromosome. So don't forget what we do today for when we come
Yes.
to meiosis.
So please, if we say it's a change in the genetic composition, it only refers to a gene mutation. So, but what can that change be? That change is in the sequencing of the basis of a DNA. And our question to you is, when do you think, if DNA, or when will a change or a mistake in the sequence of the basis of DNA occur? And there's your answer. During DNA replication, any mistake can happen. During the transcription of DNA to mRNA that we've just done, there a mistake can also occur. And then when we get to meiosis, but know these things, during crossing over, during the prophase 1 of meiosis, and then we have external factors that can also cause. You see, the top 3 are genetic changes, but an effect of radiation can cause some cancer, a mutation, or heavy metal ions. And
They say even stress, because cancer is caused by a mutation. Even if we live a very stressful life, it might cause, that is the theory, it might cause, or the hypothesis, a mutation, because it's still studied.
Okay, so we are now looking at mutations. Now, a gene mutation is a change in the nucleotide sequence. We already said when it can happen. But what does it lead to? What is the result of a change in the nucleotide sequence? First of all, if DNA provides the code for protein, for a specific protein, and that code changes, that means that the protein that will be formed will either be a wrong protein or no protein at all might be formed. So that only a change in one of the bases can lead to a lot of disorders, well-known disorders that we know today. So yes, gene mutation, and then there are other mutations as the two of us sit here. And as they sit on that side, then there are so many mutations taking place and it has no effect.
Mutations are happening while we speak. But most of these mutations have absolutely no effect on us. We will never know about them, but it's only every now and then that a mutation might have an effect. Who was first, the egg or the chicken? And there's a case of, you should be sure about that answering any conversation, because it's the egg. Because mutations occur during DNA replication, amongst other things. Yeah. So we as scientists know the answer.
So when we get to evolution, we will touch on that because it's the only way to explain the answer of which was first, the chicken or the egg. Must have been the egg because the egg contains the genetic material.
Yeah, and there was just a question. We did refer to chromosome mutations, and Down syndrome is the one that we study. So Leshwin, hold on until we do. Meiosis, we will explain it to you completely then.
And I think we're going to answer one of those learners' questions now on tRNA, how it is formed. So let's explain again. I think we've done it on the whiteboard, but DNA, once again, it provides the code for a specific protein. The mRNA must rewrite that code into a codon, and the tRNA that has a specific anticodon that matches the codon But more importantly, it carries a certain amino acid. Now, before we came here, we found a little bit of a mistake here. We are very honest.
I'm very happy I can go back to the paper. I love
But
drawing.
we're going to do it on the paper for you,
Yeah.
okay? So let's give an example. The code on the DNA is the base triplet AGC. So let's see if they can give us the codon, Lorraine. Maybe you can check our WhatsApp line to see whether the codon can come through. So
Yeah.
what will the codon be of mRNA?
What happens, do you think, to the codon and the anticodon of the protein synthesis?
Okay, let's see.
It makes yet another
Yeah.
protein.
So adenine
let's see if we've got an answer.
matches with the mRNA, the UCG. Good.
Well done. It
Wow.
is Siyabulele from Lile
So
from
what
Northern
will the
Pole.
tRNA code for? Because the tRNA needs to translate this message, because attached to the tRNA is a specific amino acid that the DNA codes for. You see, the code is on the DNA, so DNA determines that amino acid. And I think that is now they start understanding.
They're coming through.
Yes.
Through. And the anticodon, yes, you're right. Coming through is AGC.
And we have a wonderful example
We're having
here.
excellent response.
If you look here, because there is no thymine or uracil in here, that means if there's no thymine in DNA, there will be no uracil in RNA. So AGC, if you look here, RNA is actually, it's exactly the same as AGC, except when you have
there's a
thymine
U
and
a T,
uracil.
yeah. Okay.
And that amino acid that is attached to that anticodon is alanine. Let's say it's alanine. That is just hypothesis.
No, alanine. Yeah, let's
Yeah.
say, but this is alanine, and that is the
Yes.
abbreviation.
And you
What
don't
is our
need to
next
know.
series?
Sorry, you no need to know the names of the amino acids. It will be given to you.
You get a table.
There's our second one.
We have
GGC
GGC.
codes to.
Send your codes and codons.
Oh,
and
I have it on there
anticodons.
already. CCG. So let me go a bit quicker here. And it codes for valine.
Yes.
So there you see, GGC codes for the amino acid valine. And this is now the number. If you dial 021 or 012 or what, that is now the sequencing. There's a third one, TAG.
What will that be?
So they already know. There's A, U.
I can't
C.
keep up with everybody here.
Oh, they are
Ramafola
good.
Hai. Amino acid, Kunjolwa, is the building block of a protein. So let's continue. We can't do a T here. It must be a U. And then an A.
You see,
and
with the
then a
RNA,
G.
the U
Yes.
replaces the T. And that
what
is glycine.
It's glycine.
Now let's go
one of
further.
the most. This is one of the most simple amino acids.
Now you must go, Lorraine, and show what happens. Let's take that DNA GGC, because
Okay.
that is the next one.
GGC.
But there was. I should actually take that up. Just ignore those.
No,
quickly.
we will do.
Yeah.
But
There you
something
see,
happened.
Yeah, the G here was replaced by an A. It's a mistake.
Oh dear. For some reason, something happened. So it turned into AGC.
And ACG codes into, I'm actually right, I'm not wrong.
AGC will code into UC. Let's we get the board, yes please. And UCG. And it will, the anticodon will be AGC.
So just stop there. Because now, if that second one, GGC, was repeated as a fourth code, that means the amino acid was supposed to be valine.
We would
But
have,
because
yes, sorry,
the
can I
one-
just say?
Yeah.
You can see
Yeah.
it would have been valine, but no, it's not going to be. It is going to be
Alan.
AGC. It is going to be alanine. And there you can see, instead of alanine, valine, glycine, valine protein, that is your protein, it is now a different protein. it is now alanine at the end. It's a totally different protein. And this protein can either be beneficial or it can cause a terrible genetic disease. And remember, you had in genetics, you have to learn about albinism and sex-linked diseases such as hemophilia, color blindness. You also might know about muscular dystrophy, etc., etc. And all of those caused by mutations. One letter, one base. It's a gene mutation.
So I wonder if they can tell us, is that maybe clear to you all now, what a mutation does? So and there was also in one of the exams where they had to describe protein synthesis. And then, additional to that, you had to describe what the effect of a gene mutation would be on the process of photosynthesis. And we have just explained and showed you what the effect would be, because one nitrogenous base changes by mistake. It changes the whole protein. It can also be that a necessary protein, an essential protein that was supposed to have formed, have not been formed. For instance, hemophilia, that we will talk about later on. It is the lack of that protein that causes the blood to clot.
Factor VIII, which is a protein that can help with blood clotting, they don't have it because they don't have the protein, because there was a mutation. And you see, it's not that simple. There's a gene mutation on the DNA, changing a specific code, changing a specific codon, changing the tRNA, the anticodon, changing the amino acid being picked up, changing the protein. And then you either have a harmful protein, or you have a protein that cannot be formed, or you have a protein that is useless, that has no specific function. However, sometimes a mutation is harmless, but in many cases a mutation can cause severe problems. It can be useful, though, in natural selection, and as Colin has said, we will refer to that later in the year.
So let's just end off by saying, in your workbooks, you also have that little thing when you're answering questions with diagrams. There are certain things that you please need to be aware of. First of all, there's an introductory statement on the diagram. Read that. Underline each and every word. The first little word there is the action word of what the examiner asks of you. Is it list? Is it name? Is it describe? That's the most important one, because that will eventually determine why is it that you are allocated 1 mark or 2 marks for a specific question. But read that introductory statement. And then it will also tell you this is what that diagram is about. And you can look in the diagrams and all the questions in your handout, look through it, and then you must also study the diagram. If there are certain labels that you know that should be there, just write it down so that it can make more sense to you. And then look at all the questions. And that will then, and first of all, when you see a diagram, identify the topic. For instance, if you see DNA and you only see DNA nucleotides and there's nothing, no RNA, then you know it's replication and not transcription. And this is how you can identify.
So important. Yes, I agree with the one person, there's no name connected to it, but who just sent me a message and said, thank you, enjoyed, but the time is so short. We love life sciences, so we can spend hours with you. But it's been a privilege to spend this 1 hour and talking a little bit about matters in the first term. And we hope to see you soon. And we have a few more topics. In the meanwhile, work hard. It will be worth your while when you get to the end of the year.
So from us,
Keep
goodbye.
well.